Well, first off, I would like to thank the families for bringing together. What a great idea of talking about crop diversity every year and having this chat with the public. I also want to thank the selection committee for the honor of presenting this. So what I want to give you all a feel for today is how we've had this tremendous revolution in our ability to use genomics to look at DNA. And that is changing the way we look and work with plant diversity across the globe. And of course, I'm going to start with why are we talking about maize? Well, it's for a very simple reason. It's just critical for the existence of almost all of us here on the planet. Between wheat, rice, and maize, essentially those three crops support and provide most of the calories for the people on the planet. And in the US, 79% of all the grain production comes from maize. So that's why we should care. Whether you're near the farm or you're way far away from the farm, you depend on maize. So let's think about then, how maize plays a role and how, as I mentioned, this change in our ability to look at the genome is going to affect the way we make better crops. It's all through this whole process. I'll first tell you about how we're able to now read the genome, and then how we're able to go through and start understanding how genes work and how diversity really functions. And then we'll end up with talking about how we may envision inventing better crops using this new capability. And as we go through this, I want you to kind of think about those 3 steps are kind of going through 3 steps. First, we're going to learn how to become readers. We're learning to become readers of the genetic code. The second step is we're going to become scholars. We're going to learn how the genome works, how various portions of the biology of the plant work. And then at the end, I want you to put on a cap thinking about being an inventor and an engineer. How could we use that knowledge and our ability to read to do new things? So at the end of the reader section, I kind of feel like all we've gotten to is the end of first grade. We really, have spent all this time and we got to the end of first grade. We now can read a book. But I think we're also jumping really fast here, and we're also making some real progress so that we also want to be able to jump within the next few years of having finished college and start deploying this all at the same time. So this all starts with Gregor Mendel. Gregor Mendel discovered the laws of genetics in 1866. And if you think about it, it's almost been 150 years before we really now have this capability to read the genome. Part of it is because these genomes are really big. They have 3 billion letters quite often. And just to give you kind of a little feel for how this has changed through time, I'll give you a little personal tour of this. When I started doing genetics back in 1993, I would hook up solutions to various parts of the lab and would pour these gels and would then hand go through and count each base pair of the genome. And if you think about it, if we needed to go through 3 billion base pairs and still doing it that way, I'd still be doing it. But luckily, technology came along, and because of the push to sequence the human genome, by 2003, we had the tools in our lab so that we were then able to sequence nearly 200,000 base pairs per day. So that was starting to move us along. But still, 200,000 base pairs a day isn't enough to get done with the genome. And so, but what's happened in just the last decade is now we can look at nearly 38 billion base pairs of the genome per person. One person could do this per day. So it's shifted dramatically. Essentially, that's an improvement of being able to look at and read DNA essentially 25 million fold. Okay? And so let's think about how big a change it is to do something 25 million fold better. Back in 1993, some of you may have had a computer like that, one of the Macintoshes. And we normally think about computers getting a lot better through time. That's kind of our standard for amazing change through time. But essentially, that computer is, if we buy the modern Macintosh today, it's about 25,000 times faster than the one we could buy in 1993. So that's certainly a big improvement, but that's nowhere near 25 million fold better. Okay, so let's give it a feel. What is 25 million fold better like? Well, every day I walk from my house to my office, and it's about 3.7 miles. It takes me about an hour. So if I were to get 25 million fold better at walking, what could I do? Well, Being 25 million-fold better at walking would allow me in 1 hour to walk to the sun. Okay? That's how much better our ability to do genetics and to read DNA got over this short period of time. Okay? So less than a generation, we got 25 million-fold better at something. In 2009, colleagues of mine, led by colleagues at the Washington University in St. Louis, they sequenced the first maize genome. It turned out the maize genome was 2.3 billion base pairs in length. And at the time, it was the most complex genome ever sequenced. Part of this is because portions of the genome had old duplications. And there's these aspects of the genome that genes have the capability, in some species, to jump around, jump from one place to another place. You may have heard of Barbara McClintock, a famous Nobel laureate, who was the one who discovered that. But the maize genome is just chock full of these types of things. It made it a very complex genome to look at. It also had 40,000 genes, so a large number of genes. And if you think, and that took about 10 years of preparation to get to the stage where they could sequence that genome, and it cost about $50 million to do it. Now, 3 years later, we can resequence a genome for $4,000. Now, that genome isn't as good as the $50 million genome, but it's good enough for breeding, okay? And it's good enough for a lot of other purposes in genetics. And we can now profile a genome for just $15. So we went from the first human genome costing $3 billion down to being able to do some of these things for, sometimes it's pocket change, okay? So and really our bottleneck now is analysis and thinking. How do you think about billions of base pairs? It's much more expensive now to think about it than it is to generate the data. So after nearly 150 years, we got to the level that we can read. We finished first grade. So, and now what should we do once we now have this capability of reading? Well, if you're interested in crop diversity, like myself, the first thing you'd like to do is compare two copies of the same book. And that's what we can now do. We can read all those letters, and we can line up them and ask that question of how similar are they. So it's always good to have a frame of reference for this. And let's start out with how different you and I are from one another. So you and I only differ from one another by about 1 in 1,000 base pairs. Doesn't matter who it is in here, any one of us, we only differ by about 1 in 1,000. Now, if we compare that to chimpanzees, they're about 15-fold more divergent than you and I are from one another. Now, how divergent do you think two corn varieties are from one another? If I took a Monsanto corn variety versus a Pioneer corn variety, how different do you think they would be? I think that they probably look to most of us like they're 100% identical. Turns out that doesn't really matter what two corn varieties I take. Any two corn varieties are more different from one another genetically and at the DNA level than humans are from chimpanzees. And as I'll show in a few seconds, it's actually even more extraordinary than that. The reason we have trouble telling the difference between two corn varieties, but we have no trouble telling the difference between humans, is because we're evolved to differentiate between people. We're not evolved to see the small differences between the corn plants. So what does a human genome look like? Essentially, this is a little snapshot. Remember, the human genome is about 3 billion base pairs in size. But this is just a little snapshot of 100,000 base pairs. And in that region, there may be 2 genes. And that may be me, and this is you. And essentially, there may be 100 base pair differences along that 100,000 base pair stretch. Maybe a small deletion. And those 100 base pair changes are enough to make the differences between you and I. Now, let's compare what would happen if we were to line up 2 similar segments of the corn genome from 2 different varieties. Essentially, it's dramatically different. Yes, you still have those 2 genes. They're kind of in the same basic order. But now instead you have 1,300 changes that a single base pair changes have gone on. There's whole chunks of the genome, deletions, insertions have changed. Jumping genes have moved around and reoriented things in between each of these genes. And then not in every 100,000 base pair segment, but fairly frequently, there'll be a new gene. So variety 1 will only have 2 genes there. Variety 2 will have 3 genes there. And this is just the makeup of the genome. There's far more variation of what's between any of these 2 varieties. And when colleagues of ours have looked very closely at two inbred lines and just asked the question, I'm going to line them up end to end, how similar are they? It turns out to only be about half the genome is at the same position and inherited from parents in the same way. In comparison to humans, it's 99% the same. When I talk to my colleagues in medical genetics, I often give them a hard time and they're studying diversity. I said, there's no diversity there. There's nothing to study. But obviously there is, and it's quite interesting. But we do have a magnitude of difference between what's going on with some of these crops. And it really always strikes me, how can two corn varieties really look the same when their genomes are so different from one another? And it comes back to the fact that essentially this diversity is very old. Plants, and maize isn't really that exceptional in this regard, having a lot of diversity. Many plants are very similar to this. They have large populations. There's, at any given time right now, actually there's 10 trillion corn plants grown on the planet. And throughout most of its history, it probably had billions of plants were growing at any one time. The humans only got to a billion a few years ago, or 100 or so years ago. And so most maize diversity doesn't have anything to do with the domestication of maize. That only happened 10,000 years ago. Most maize diversity actually goes way back. It goes back millions of years. Most of it's in this kind of 1 million to 2 million, but there's a little bit of the diversity that probably goes all the way back 4 million years. It evolved in a time before its sister genus even diverged away from it. And when climate was different, North and South America weren't even quite attached the same way they are today when some of that diversity was starting to evolve. In contrast, human genetic diversity only goes back 20 or 10,000 generations to about 200,000 years ago. And so the reason why most humans look genetically look the same is because of that short time frame. There just hasn't been enough time for diversity and adaptations to evolve to all these numerous environments. So let's start talking. So we got to this point where we can now read the genome, and we can start comparing a genome. Now let's see if we can start understanding the function of the genes that are in there and try to understand what's the diversity out there. Let's, 10 years after Mendel made his discovery, Charles Darwin, he was already playing with corn plants. And he actually made this comment after having done a small experiment, that cross-fertilization is generally beneficial and self-fertilization is injurious. If we were to put it in a different way, hybrids are good, selfing is bad, or inbreeding is bad. That's essentially what he was saying, and the reason he said that is because he took either outbred corn plants or self-corn plants, and he grew them in a little pot and measured how tall they were after a number of weeks. And this is his data, and as you can see, the outbred corn plants are taller than the self-corn plants are. At the time, he didn't understand the genetics to make sense of that finding, but essentially that finding is what gave rise to the hybrid corn industry. That essentially is now what essentially all the food that we get from maize is today. But also it sets up a very important biological question of why do hybrids work? Why are hybrids better than inbred versus, or why is selfing bad? Why is outbreeding good? And so we can now rephrase this question that's almost 130 years old in terms of now the genome. And we can now look, because we can now read, we can ask the question, can we make sense of this observation through our ability to read? So before we go down some of those questions, I want to make sure people understand that just because we have a genome sequence, it doesn't give us real knowledge. Essentially, it's like getting all the letters out there, and maybe it's, I don't read Italian and everything. It's kind of like looking at Italian. There's maybe some words I can recognize and say, oh yeah, that kind of looks like something in English, but I don't really understand it until we analyze it against something else. It doesn't tell us what are the functional variants in the genome. We can only guess at probably about half the genes, what they do. And those come from comparing them to, say, things like yeast and other organisms. But even those guesses are rarely accurate enough to be useful for breeding. And the other problem is that plants are surprisingly complex. If you asked people 20 years ago how many genes humans would have, they would have told you that humans have far more genes than plants. You know, we certainly feel like we're much more genetically complex than a wheat plant is or a maize plant. But it turns out that yes, humans are more complex than flies. Flies have about 15,000 genes, and humans have about 20,000 genes. But you take little weeds, barley or maize, and they have far more genes than humans do. And at first thought, this may be really kind of surprising. But what I want to suggest is the reason if you think about it, you know, at least the hypothesis I prefer on this is that animals have this great adaptation. When things are bad for them, they go run away. The environment is bad, you know, something's eaten it, or they get a pathogen, they go run away. So essentially, they try to maintain, through mobility, they try to maintain an environment. Plants don't have that choice. They have to live with whatever environment is thrown at them. They have to withstand the rain. They've got to withstand the diseases that get thrown at them. They've got to essentially take everything because they're situated there in the ground. And that may be actually physiologically more complex than being an animal. So, you know, whether or not that's the right reason or not, corn still has more genes than humans do. So the question is, how do we start relating this complexity? How many genes and how many variants control things we care about? Like, you know, I may want to manipulate when a corn plant flowers, or I may want to improve the level of provitamin A in corn, or be able to pack plants tighter together. How do I do that? Well, the first thing we do is we cross plants. And then we put out thousands and then millions of plants out into the field, and then we measure them. And unfortunately, measuring hasn't gotten 25 million-fold faster. It's still really hard and it's really sweaty, and you don't see me in my suit when I'm out there doing that. So, but it's lots of fun. I hope it does become 25 million-fold faster someday, but right now it's still not. But we have to go out and measure a lot of corn. And then what genetic mapping is all about is we take that knowledge of where the genes are, and we have now these catalogs of millions of genetic variants because we've taken and lined up these genomes against one another, and we know about hundreds of millions of variants across the genome. And then we take those millions of plants we've grown out in the field, and then we assay them under drought conditions, or we assay them under different flowering regimes, where we measure when they flower, we ask what their yield is. And then essentially we do a series of statistics and mathematical analyses to essentially start drawing the lines between them. And of course, there's lots of lines. When you've got 200 million variants affecting all these different traits, you know, we're talking about hundreds of thousands of relationships between each of these. And it turns out that there's probably on the order of every single gene in maize probably has some functional variation. And in total, there's probably hundreds of thousands of those lines one could draw. And perhaps even a million or so lines of them are functional in doing these. So for those young people who are out there looking for new careers, it's actually really cool to do this, okay? We're still we need lots more people to do this. And you know, just think about the numbers we get to play with. Nearly 100,000 different varieties. We have 36 trillion data points that we're trying to relate. And you know, so there's lots of really fun math and computer science to be done. So for those who are looking for exciting fields to go into, this field certainly needs more people. So now let's see what we can do in using these tools we have. Let's start out with something that's relatively simple. This is the provitamin A content of maize. So there's millions of people, or about a 250 million people around the world who are vitamin A deficient. And my colleague Torbjörn Rochford really introduced me to this, and we've been collaborators in this project throughout. And the problem is that essentially we'd like to biofortify or essentially increase the vitamin levels in the maize that people eat who are deficient in vitamin A. And as you can see, maize varies from everything that's pretty much white all the way to something that looks like a carrot. But the problem is that you could say, well, just tell people to eat orange corn. That would solve the problem. Problem is, orange corn doesn't necessarily have a whole lot of beta-carotene. So the shade of yellow doesn't really correlate with beta-carotene content very well. And so in order to figure that out, it actually takes an assay that costs about $50 to figure it out, and a lot of work, and not really doable on the scale of looking at hundreds of thousands of varieties. It'd be a lot better if we could just look at the genes that were underlying that and figure out why one variety has more beta-carotene than another variety. And so that's what we did, is we first phenotyped all the plants. We asked how much beta-carotene is in all these different varieties, and it turned out there was 66-fold variation in beta-carotene levels. And then we looked at every single gene across this pathway and asked, are there natural genetic variants that allow us to increase these? And it turned out there was 2 genes that if we put the right 2 genes and the right 2 allelic variants together, we increase the level of beta-carotene 16-fold. So we're not using, these are all the natural variants, but just had to put the right 2 together. And the key thing was the right 2 variants weren't in the same germplasm pool. The one breeder could have probably gotten one allele, another breeder working in another part of the world would have been able to figure out another one of those variants. But it was because we put the genomics together with the knowledge of the biochemistry that we said, put these 2 alleles together and pop, comes out 16-fold increase in beta-carotene. The second thing is, I showed you how much corn is being grown and how important it is. The other thing about each of those are also locally adapted. There isn't one variety of the highest yielding corn. There's 1,000 varieties, or actually probably 3,000 or 4,000 varieties of corn that are planted every year in different regions of the world. So every time you want to find something, a great new genetics out there, you actually, if you want to go start using it, you've got to deploy it to 3,000 or 4,000 other varieties across the globe. So you want to make each of those steps as cheap as possible. And by figuring out the genetic basis of this, we made it 1,000-fold cheaper to essentially move the genes that allow us to have higher levels of provitamin A. Now, sometimes that's not enough. Sometimes we need to grow more. That study was just done with a few hundred lines that we looked at genomically. Sometimes we needed to grow a lot of corn plants. And so a number of years ago, in a big collaboration, we helped create a big platform where the whole public sector could work together on capturing a lot of maize's diversity and then asking the question, can we use it to dissect these important traits? So we crossed lots of lines, created lots of lines from that, 5,000 lines, and then we've grown millions of plants in environments all over the globe. And essentially, it's now become the largest genetic dissection system essentially ever created. And one of the traits we've taken a real deep look at is flowering adaptation. Just as I mentioned, we want to move genetic variation from one part of the world to another part of the world. Unless we understand how those plants flower, we can't move things between latitudes. And maize varieties vary from everywhere from about 45 days of flowering to 125 days of flowering. And maize, of course, grows everywhere from South America all the way to Northern Europe. So there's just a tremendous amount of flowering time variation. But when we looked at the genome and we asked the question, how many genes control this? Well, more than 50 genes control the trait. And the surprising thing is, despite the fact that some plants flower in 45 days, some in 120 days, the biggest effects we found at any one gene is only measured in about a day and a half. Nothing has a big effect. Everything is modulated by lots and lots of little effects. And so how do you end up with plants that flower really differently? Well, early flowering plants have lots of genetic variants that make them flower early, and late flowering plants have lots of genetic variants that make them flower late. But because we have that model now, we now understand that, it allows us to start tapping into and rapidly being able to tap into genetic variation from everywhere from Mexico to northern South America that we can start applying to some of our Corn Belt work, or we can take Corn Belt varieties and apply them to sub-Saharan Africa. One of the traits that if the farmers out here, they certainly have recognized this, that one of the traits that's really changed over the last 100 years in maize through some fabulous breeding is how closely we can pack the plants together. This is kind of the density that it used to be done at, and this is the density we now plant them at. Essentially, the plants still produce ears that are roughly the same size. So even though they're now spaced, say 6 inches apart, 8 inches apart from one another, they're still putting on this massive yield. And that's because of good breeding. And part of that was that the breeders changed the leaf morphology. There's probably a lot more. And so we thought, well, this is a change that occurred over the last 100 years. How complex could this be? Well, it turned out it's the same story as flowering time. It's lots of genes, 30 or 40 genes. And we've got maize leaves out there that are like this, all the way up to something like this. And the biggest effect changes it by 2.5 degrees. So, I mean, that's like that. Okay? So the genome is really complex. There's lots of small effects out there. And that's just kind of the way it is. We looked at plant height. We've got plants that are 3 feet tall to 10 feet tall. Biggest effect is 1.5 inches. Okay? I mean, again, lots of small effects. So at this point, if I were you, I'd be pretty depressed that everything is going to, tiny little effects and there's nothing we can really do about it. But it turns out, if you know how to do the math right and you add up all those little effects, you can predict things. So if I add up all the predicted effects of flowering time across all those genes, this is our predictions and this is what we observe. We can get some really good correlations. And we can do this for other traits. Here's height. Height's even more complex. We add up all those little effects, and they do kind of all add up, and you can make accurate predictions. So by characterizing the genomic diversity well enough and getting down and scanning all three, nearly 2.5 billion or 2.3 billion base pairs of the genome, we can start making useful predictions. So let's go back to that Darwin question of why does hybrid vigor exist? And just for, if you all haven't seen how big the difference in hybrid vigor can be, these are 2 inbred varieties of maize that you cross them together and it produces the one in the middle. And it produces this ear. I mean, sometimes the yield between the hybrids and the 2 parents can be 2-fold, 3-fold different. Just massive differences in them. Mathematicians and quantitative geneticists, going back to the 1930s, had great hypotheses on what was going on. But essentially, they really couldn't test them rigorously. And so with our new knowledge now, we can go through, look through the genome, and ask that essential question. And so in the last few years, we've really been able to answer Darwin's question of what is hybrid vigor. It turns out that hybrid vigor occurs because bad mutations happen all the time. Every time an organism reproduces, there's maybe 40, 50, 90 90 mutations that occur, and a certain proportion of those are bad. In the regions of the genome and you've got to get rid of those bad mutations somehow. And the regions of the genome that mix very well, or what we call recombine, you're able to get rid of the bad mutations fairly rapidly. In the regions of the genome that you can't mix very well, essentially what happens is you need to be hybrid. You need to have a copy of one chromosome that came from one parent and one that came from somebody very different, because essentially the 2 sides are covering for one another. And so now we know molecularly that essentially this is the basis of hybrid vigor. So, and that of course also allows us to now start making predictions. What 2 varieties of maize should we put together to make a hybrid that's going to perform well? So this is just a snapshot of the traits that people are kind of working on. People, of course, are trying to work on everything from nitrogen use, do we get bigger ears, how do we capture more light, how do we fight off all sorts of diseases, how do we improve the oil content, and so on. So there's lots of areas that now can be attacked with these tools that people have built over the last few years. I also want to highlight, some of you may have heard about personalized genomics and human genetics right now. And essentially, I'm really hoping that it'll become a great thing, but it's likely to happen much slower than what happens in plant genetics. The big problem in human genetics is you can't organize 1 million people to all grow in a field together, and they aren't willing to allow you to poke them and measure their DNA. But in plants, we can do that. And that gives us a tremendous power. Now that we can read, we can really read, we can evaluate how plants grow much better. The second thing is, as soon as we find useful variation, we can use it in breeding. In human genetics, it's often just the beginning of a search for a new drug or a new therapy. In plants, of course, we can just use that immediately in agriculture. And so we're going to see some of the benefits of this genomics revolution in plants actually far before we do in human genetics. So as we start going down this arrow here, let's start talking about let's start envisioning what better crops would look like. I think what we're starting to get to is we now know how to read and we know how to understand. We haven't read everything, we don't understand everything, but we kind of have a guidepost of how we should go about learning these things. And I think what we should start really asking ourselves is, what should we invent? I feel like we're kind of in the late 1800s in what Thomas Edison might have felt like, when electricity was finally harnessed. And essentially you have you have an opportunity for a massive explosion in a number of inventions. In genomics, we now can do those first couple steps. And now what should we, as both geneticists and society, what do you want us to invent? It should be a dialogue between all of us of what is possible and what should we invent. So what can we do here? Let me start out with, I don't want to act like there was no invention in the 20th century, because maize breeders actually did a remarkable job in the last 100 years. And the way they did it was they made crosses, they inbred those crosses, they then came up with very clever ways for starting to trial the plants out in the fields in organized fashions. Once those worked, they advanced them to larger scale trials, and then the ones that won those trials, they helped invent a lot of the statistics we now use today. Turned out that you could then pick out the winners, and then they made new crosses. And you essentially went through that cycle. Every 5 to 6 years, you went through that cycle. And it worked. And it really worked. If you think about starting back in 1865, these are corn yields in the US. They're essentially flat until hybrid maize comes about and people really start going through that cycle of using genetic variation, organizing it, and cycling through it. And since then, corn yields have gone up 8-fold in the last 80 years. Half of that is due to genetics, and half of it is due to changes in agronomic practices, things like increases in fertilizer. But it's been a remarkable change, and so that cycle absolutely does work. And I want to make a point that productivity of crops also matter. In the US, back in 1900, we had those low yields in maize, and a lot of that low-yielding maize was essentially occupying land throughout the whole Northeast, Virginia, and of course the Midwest. And essentially, where I live, there were no trees left. Near the gorges, there was a few trees left. Everything else was under cultivation. Just because the yields were so low, in order to support the population, they had to grow corn everywhere. Because of good genetics, essentially we were able to move maize agriculture to the high-yielding Midwest, and essentially the Northeast is now reforested. So essentially, that whole area now has my area is like 50% increase, but across the whole area, it's about a 16% increase in forestry because of productivity gains in maize. And of course, it's radically changed, agricultural life. At the turn of the century, half the population worked in agriculture, and now, because of these gains in productivity, we all can learn about agriculture. We can do agriculture if we'd like to, but it isn't required that half of us do agriculture. It's allowed us to have a lot of the other things that we enjoy in life today. So, what does variation can do in genomics? It allows us to evaluate natural variation. If we can then use that to mathematically model how we can predict, then allowing us to predict things, and it facilitates rapid breeding. Let me give you a little feel for what some of these key aspects are. I can look up on various indices 100,000 different varieties of corn, but which one should I cross? I only have, that's millions of possible things I could cross, but what should I cross? I don't have time to test everything. There's not enough land on the planet for me to cross every variety with every other variety out there. So by having genomics, it allows us to start getting guesses at what we should cross. The other big change, as I mentioned, it can cost $15 to do a genomic profile of a plant. That's cheaper than it is for me to organize putting those plants out in the field to measure them. So it's totally changed the way we think about doing plant breeding. It's cheaper to look at the genome than it is to grow the plant. So we now try to figure out how do we genotype everything we possibly can, and then we do the expensive part of putting plants out in the field. And the other key thing is we now know what those 2.3 billion base pairs are. And we have an idea of where we'd like to go. And essentially, it's becoming more of an engineering question. What's the fastest way for me to organize those 2.3 billion bases across the genome so I can produce the crop I want? So we had that, I showed you that model, the standard model of breeding. This is what genomics-based breeding has the potential to do. Essentially, if you grow corn in Hawaii, every 4 months you can harvest it, plant it again, and grow it again. And that's what all the major seed companies do. They are essentially turning 3 generations a year in Hawaii and essentially going through that type of cycle. If you can predict, take some mathematical model and are able to predict phenotype well enough, you can actually make gains that are as good as this 5-year cycle. So theoretically, we could essentially breed at 15 times the rate that we were doing during the 20th century. Now, we realize that, that we can't, that's the biological maximum for doing this. There are certainly imprecisions in our ability to engineer that genome and everything. So we don't think we can get to 15-fold. But the question is, how far can we push genomics and genetics so that we approach that biological maximum? And so we have these tools, and then this is where I think the question is, what should we invent? What societal problems should we take on as we create a next generation maize plant? Should we make it more nutritional? Should we make it more drought tolerant? Should we make it more fertilizer efficient, higher yielding, and more disease resistant? I'll talk about the first 4 topics. I know a colleague of mine talked about disease resistance a couple years ago, and she knows far more than I do about it, so I won't go into that. So let's talk about biofortification. This is essentially why we got into making high levels of provitamin A in corn. But the basic idea is let's add vitamins and minerals to staple crops. Why is that important? Well, there's a lot of places in the world where there's no way that there's a food chain or a supply chain where we can fortify the wheat the way we do here in the US. You need to do it biologically because there just isn't the transport in large parts of the developing world to provide fortification through other types of approaches. People will also ask, well, why would you want to do it when people should just eat vegetables? Well, that's a great idea. And I'm all in favor of eating lots of vegetables, but in lots of arid regions, you only have fresh vegetables the 4 months of the year that it rains. The rest of the time, people are having to live on stored grain supplies. And where do people need to get their vitamins at that time of year? And so that's where fortifying our staple grain crops is a really nice way to supplement the vitamins that we get through other sources. And some of the targets that people are aiming for are high levels of vitamin A, zinc, and iron. We already mentioned the vitamin A deficiency in helping out so there's about 250 million people around the planet that are essentially vitamin A insufficient. And that causes a lot of immune issues. And then there's about 1 million children a year who are going blind because of low levels of vitamin A. In the US, a lot of macular degeneration is the product of not having enough zeaxanthin and lutein, which are also carotenoids. So these are all things we can manipulate in maize to help out both these types of problems. But it takes the spirit of an inventor. My colleague, Torbert Rochford, he's got that kind of spirit of an inventor. Once we figured out what the genes were, he said, great, I'm now going to go work with my colleagues at CIMMYT, and we're going to go see if we can push this out into cornfields in sub-Saharan Africa. And so he teamed up with a really great institution called CIMMYT. They're headquartered in Mexico, but their goal is to develop better varieties of better maize varieties for the developing world. And they support national breeding programs throughout the developing world. And one of the targets they're now working on is trying to improve the nutritional quality of maize. So Torbert teamed up with Kevin Pixley there, and just in a couple years, I mean, it's only been since in 2008 when we first reported this discovery, but in 4 years, they've been able to take that genetics and put it into varieties that are adapted into Zambia. And this year, people are trying out new varieties of these corn that have high levels of provitamin A. These are about 3- or 4-fold higher levels of provitamin A, and in 2 years from now, there'll be that variety that'll be 16-fold higher levels of provitamin A. And at that level, it should really be able to help out a lot of people with vitamin A deficiency. But it took that spirit of invention that Kevin and Torbert had and the tools of genomics to really move this forward. Let's talk about drought tolerance. Of course, everywhere in the Midwest got hammered by horrible drought this last year. Maize crops were failing. Indiana, the yields were incredibly low. On this, I'm not quite as optimistic about our ability to deal with drought tolerance genetically as I am some other areas. But I'll give some strategies here. The key problem is you can look across the globe. Where do plants do the best? Where do they photosynthesize the most? Where do they make the most carbon? Well, it's where you've got lots of heat, lots of sunshine, and lots of water. You really need water in order to do photosynthesis. And this is a plot of annual average precipitation versus net productivity, and you see how tight that line is. There aren't environments that really break that line. You do need water to get plants to grow. So unless there's some bright person out there in chemistry who can really help us figure out how to change the basic chemistry of how plants swap water for CO2, what we can do genetically is help plants get through short periods of drought. We can get through a period of, say, a month of low water and things like that. We can certainly genetically improve our ability to withstand those types of things. It's not clear to me there's much we could do in Indiana, but we probably genetically could have done a lot better in, say, Iowa this year with our corn crop if we had even more drought-tolerant varieties. But I would highlight that our corn varieties, the seed companies and a lot of people have already been working for a number of years trying to make very drought-tolerant corn. Actually, they did a very good job in making drought-tolerant varieties for Nebraska. And the reason why the whole corn crop didn't fail is because actually a lot of work had already gone into it, and it prevented it from being a total failure this year. One of the areas where I think genetics can do a tremendous amount is in reducing the amount of fertilizer that is wasted on our crops. Only about 50% of the nitrogen that is applied to our crops actually is taken up by them, and only about 16% of it actually gets into the people downstream, and instead, most of the rest of it goes down the Mississippi River, and you're seeing the algal bloom that essentially is in outside of Louisiana because of the fertilizer. And of course, this is a waste for everybody. It's an environmental hazard. Farmers don't want to waste nitrogen. Nobody wants to waste the fossil fuels that it took to produce that fertilizer. And there certainly is a lot of genetic variation out there for nitrogen use efficiency. And I think there's opportunities between different changes in agronomic practices and better genetics. We can make real progress there. And just as I was telling you, I love sequencing corn. Well, other people out there actually like to sequence the dirt. It turns out there's microbes everywhere you walk, and a lot of those have the ability to manipulate nitrogen and do all sorts of other things. And so in the next decade, there's probably likely to be a lot more synergy between what's going on the soil beneath your feet and the roots of our plants, and we'll probably be working together to try to use genomics to figure those things out so that we can make some of the biological fixation of nitrogen. One of the other things that a lot, both companies and people in the public sector are aiming to do is to just basically double yield. You know, we've already shown you how maize yield has increased 8-fold in the last 80 years in the US. And the reason we think we can keep on going farther is every year they hold a yield contest in maize. And it's always amazing, but it's pretty much always up here. Near 20,000 pounds of corn are produced per acre. I mean, that's still an extraordinary, I mean, the pile of corn that can be produced in 1 acre is amazing. And everybody can kind of complain about corn taking over too much acreage, but the thing is, it produces a lot of corn per acre. It's a very impressive photosynthetic plant. And so US average is down here. This was the horrible year this year when the drought hit. But see, this is our bad year. These are the average years for East Africa. Okay, we're talking 5, 10-fold less sometimes than even our average years. And some of that has to do with not having enough fertilizer, but a lot of it also is putting out varieties of maize that are maladapted to their local environment. And so through a combination of different approaches, we can certainly double these yields. And I think there's a reasonable opportunity that perhaps with the right genetics, we can even double US yields. You know, but it's going to take a multi-pronged approach. It requires increased levels of heat, water, and drought tolerance. And there's also a tremendous variation between plants in the exact same varieties in the exact same field. You'd be shocked at how a plant right here and one right over there, that one will have half the yield the one does over here. And so a lot of being able to increase the yield on our crop plants is to try to figure out how do we get everything even everywhere and all up to how the top 90% are performing. So of course there's also opportunities for better agronomics to deliver some of these things. And you know, as I mentioned, I think there's a tremendous opportunity in the developing world to do a lot better. So I want to end with a thought-provoking idea of, you know, what is a more crazy invention? You know, we in the maize community are all going to do the things I kind of suggested there. But the question is, could we solve 3 of these problems all at once by making maize perennial? And let me put forward the argument on why we should at least entertain this idea. So I kind of view this as the road not taken. You know, and it's kind of a strange thing that's not taken. Most plants in the world are perennial. You go walk out in the forest, of course everything's perennial. Almost everything's perennial out there. You go walk to a prairie, almost everything is perennial. You know, and even most domesticated crops are perennial. 61% of all domesticated crops are perennial. The problem is the top 5 crops out there are all annuals. And, you know, and there's a major cost to that. But we're not necessarily stuck there. Every major annual crop out there has a perennial relative. Cultivation of all annuals and perennials generally works pretty well. I think the main reason we ended up with annuals, especially for these top 5 crops, has to do with that annuals respond to selection faster. So you can imagine 8,000, 10,000 years ago, if you're a first farmer and you're trying to breed various crops, it's a lot easier to work on a crop that responds to selection every year. And so they were trying to breed in certain ways, and of course the annual was the one that worked. They burned the environment, the annual was the one that came back, and they were able to improve. And so we kind of got stuck with these annuals because they were grains, because they're the one that respond to selection, not because it's necessarily the best physiological approach. But I think it's time to revisit this question. Genomics can, you know, accelerate breeding even faster in certain regards for perennials than annuals can. So, and then what's also struck me is there's been lots of biofuel research done in the last couple of years. And a colleague of mine, Steve Long, he carried out a study in prime Illinois corn real estate. Beautiful cornfields out there. The corn doesn't do any better than in these fields. And what he did is he planted maize and he planted miscanthus. And this is actually a distant relative of maize that's used for biofuels. And it turned out that miscanthus fixed 60% more carbon than maize does. And the reason it fixes 60% more carbon is really simple. It grows longer. You know, it collects the light, it uses the water for more time than corn does. You know, here's the timeline. essentially miscanthus is jumping up to being a very productive plant, and it's got a month and a half, 2-month head start on, and it also grows more at the end of the season. And it does that every year. And essentially, it's using a lot more of the light and water that's available. Let's look at this on a continental scale. So the green here represents the regions of the US with high levels of photosynthesis at the time. And here we are at the end of March. You can see the south is starting to light up. But what you see, and follow this essentially photosynthetic desert right there. So now we've moved to the end of April, and you still see that photosynthetic desert. You know, even New York, where we are, where it's cold and things like that, we have more photosynthesis than out here. Now we're at the end of May. There's still a photosynthetic desert sitting out there. Every place else that has forest cover is making lots of carbon. But it isn't until we get to the end of June that that photosynthetic desert is filled in. And I'm sure it doesn't come as any surprise what that photosynthetic desert is. That's the Corn Belt. And essentially, that Corn Belt isn't producing photosynthetically until after the summer solstice. Half your good light is already gone. You know, and it's because we use an annual plant on that space. So what would be the benefits of perennializing our crops? You know, one of the ones that's long recognized, colleagues of ours at the Land Institute have shown this is reduced soil erosion. Better fertilizer use efficiency. Once you've got good root systems and you stabilize the land, nitrogen doesn't seep off it very much. You know, because you have a deeper root system, your drought tolerance is better. You're not going to produce if you don't get any water, but you can get through those 1-month periods without water better. And I would argue that we have an opportunity for higher yielding through a longer growing season. And for environmental purposes, there's also the possibility of sequestering carbon in a perennial far better than one does in an annual. The concerns that people have raised, one is certainly a lot of modern perennials, they put a lot of energy into their roots. But that's a selectable trait. You know, a breeder could decide exactly how much carbon and how much root maintenance goes on. We don't have to have roots that go down 35 feet. We can have roots that are stable and shorter. That's a selectable thing. There will be disease and pathogen pressures. It will certainly, as Tom and I were just talking, there certainly will be lots of new pressures. But there's perennials already out there that dominate ecosystems. They're able to deal with their pathogens. Yes, it'll certainly provide job security for people in those fields, but it's something that can be dealt with, I'm sure. Weeds and stand maintenance, those are things that essentially, yes, you may lose some and we may be more tricky to keep up density, but I've already just heard about a robot that goes through and essentially weeds lettuce fields and is just being deployed. So I think if we're talking about 15 years from now, the ability for us to, in a mechanized way, deal with the weeds and send a robot out there to go deal with it is probably not a big deal. And the question is, would there be a market, and then would there be a value for people to produce types of seeds like this? And I think even the seed industry would like to get into this, because essentially they spend about 1/3 or 1/4 of their cost in actually making seed. So they could essentially sell the seed for 5 times as much and use it for 5 years. Because people would want to probably swap out for the newest, better perennial every 5 years. Or if they were doing rotations, they'd probably want to rotate every 5 years or so. Just to show that biologically, the switch between perennial and annual isn't rare. This is the Andropogons. These are some of the most efficient photosynthesizers on the planet. With things like sugarcane being right up there, miscanthus right up there at the top. But these are all closely related series of grasses. And what's happened is in all these different, across all these grasses that are closely related, the annual habit and the ability to move from, having moved from temperate or from tropical to temperate has happened over and over again. I even think it may be a simpler trait than perhaps some aspects like flowering time and some of these other ones. But it certainly is genetically possible to make an annual out of a perennial, and I'm sure we can reverse it. So what some colleagues and I are starting to work through and think through is this type of thing. Can we cross maize with Zea diploperennis, its perennial wild relative, and find the perennial genes? Can we cross this sister genus that is adapted all the way to northern areas as far north as, say, Massachusetts and northern Iowa, and cross it with a southern variety of that and clone the cold tolerance genes? If we were able to figure out what these genes are, we could then essentially lay the groundwork for making a perennial maize variety. So then we get to the question, if we can find these genes, and I'm sure we can find the genes, it just takes the resources to put those together, how would we then use those genes to make a perennial maize variety? One possibility would be to cross these into an elite variety, and we could do that. But these are some, especially that sister genus, it's a little hard. We can cross them, but it's hard. And it would take a while to get it to work. And then the whole problem is, just as I mentioned before, there's thousands of corn varieties out there. If we want to make hundreds of perennial varieties out there, we'd need to do this over and over and over again. So I think the better strategy, rather than doing regular crossing, is to use some of our new genomics and genetic tools of let's make an artificial chromosome that captures the best genetic variation from, say, Zea diploperennis, and put that on a new chromosome. All the genes that make a perennial, stick them on a new chromosome. Take the genes that provide cold tolerance, put them on a chromosome. And then all we have to do is move that one chromosome into each of our annual varieties of maize in order to convert that. So that's the dream. I don't know how well that would work, but I think the question is, should we try these types of things? And is it worth it? So is it genetically possible? I think the answer to that is definitely yes. All the wild relatives are perennial. Can we do it? Yes. It just takes resources and time. Will it be better agronomically? I've provided some arguments that yes, it would be better agronomically, but I can't prove that. And so, like in many areas of science, we have a hypothesis. And then it really is the question, should we go ahead and test this type of hypothesis? Are the benefits large enough that it would be worth to go test this type of hypothesis? So I would argue that it probably is worth finding it out. And I would just want to highlight that just in the U.S.'s corn crop over the next 20 years will be worth, combined over those 20 years, will be worth about $2 trillion in today's value. It would probably cost $100 million to make a perennial corn variety. And most of that would actually be the cost that would be in the latter development phase, once you already knew whether or not it was going to kind of work. These are the type of, and I would say that cost-benefit analysis is probably a risk worth taking, because it would certainly dramatically change everything about how corn production and the sustainability of corn production is. And I don't know that this is it's my personal favorite idea, but there's lots of other great genetic inventions that I think people should start thinking about and envisioning. And we should try multiple of these. Maybe this one fails, but maybe we should spend a few of these percents on 5 or 6 different great ideas that would really change the way we do agriculture. So I think now is really the time for genetic inventors. But I think here we need to start asking the question, what should we invent? In Edison's time, people wanted to have recorded music. They wanted to see pictures of people from all over the world. Those are great things. So what does society want those of us who are geneticists and breeders to do? What are your goals for us? And we're these tremendous inheritors of genetic diversity that's come through the last millions of years. And we now have the ability to understand it and use it. And so let's try to think about how we can use it thoughtfully. So I just want to end with, I have lots of colleagues across the country have worked together on this project and on these ideas. And I just want to highlight that everything I've talked about required a whole team of people to do it. And this work was supported by the National Science Foundation and the U.S. Department of Agriculture's Agricultural Research Service. And I'd be happy to take some questions.